Welcome to Study Loop.
For this question, we're looking at the codon and how this is interpreted to make different amino acids or parts of a protein. So the first part of this question, we're looking at different experiments by Crick and his associates, as well as experiments by Nirenberg and Mattei. So the first experiments by Crick and his associates were dealing with adding mutations to different DNA sequences inside a virus. So you could imagine a virus could have a DNA sequence such as this of ATA, GCA per se. What they did is they're going to add in a different number of mutations to see what effects it would have on the virus and its capabilities. So they noticed that, of course, the unmutated virus had a normal function. Two different viruses with either the addition of one or two nucleotides showed no correct function, and the virus that picked up three nucleotide additions also had a normal function. And this was used to prove the importance of three nucleotides to make up one codon. This is because in the addition of either one or two nucleotides, it's going to cause a frame shift mutation. And this is because three nucleotides are required to make the protein. So when one nucleotide is added, it's going to pull two nucleotides from the next three pair of nucleotides. And that's going to alter all the next upcoming proteins, and it can severely alter the capability for the virus to function. This is the same for the addition of two nucleotides because it's still going to grab the first nucleotide of every next amino acid grouping or codon coming up. However, the last option appeared to be normal because with the addition of three nucleotides, this would only correspond to one mutated protein inside the virus. And typically, one mutation of a protein is not enough to completely alter and destroy the function of the cell. So the rest of the amino acid groups or codons located later in the DNA sequence would still be normal, allowing for the virus to function normally. As for the experiments by Nirenberg and Mattei, they discovered that with the addition of ribosomes and, say, a certain amino acid, if they only had, say, U's or A's or G's inside of a solution, it would only create one type of amino acid. Whereas the addition of different nucleotides and amino acids would correspond to a completely different type of amino acid. So that just helped prove the specificity of the coding sequence where one type of codon is going to produce one type of amino acid as long as the RNAs or ribosomes are present inside the solution as well. Moving on to part B of the question, they're asking us to calculate ratios, or the percent chance of different combinations of codons. So we have three different types. We have the first scenario where we have two guanines to one cytosine. We have one guanine to two cytosines, and we have the last scenario where we have only cytosines. And of course, the ratio of G's to C's in here is 5 to 6. So 5 guanines for every one cytosine. Now you can add these together to get the whole of the probability chance. So this would be equal to 6 parts to the whole. When calculating this inside the problem, they make it a little complicated, but it's just calculating simple chances, and probabilities for each of the amino acid sequences. So we'll say we're calculating the ratio of this codon, GGC, to the percent chance of randomly coding for all guanines. So the chance in this solution of coding just for a guanine is going to be 5 out of 6, because pulling 6 times from the solution, 1 time you're likely to get a cytosine, whereas 5 times you're likely to pull the guanine. So again, for the second guanine, you have a 5 out of 6 chance. Whereas for the last nucleotide, the cytosine, you have a 1 out of 6 chance, or probability. And then they are just dividing this by the chance of getting all Gs in this other codon. So again, the chance of getting a guanine is 5 out of 6. So this is all they are doing in the problem. And then they are just simplifying it and rewriting it as, say, 5 over 6 squared times, say, 1 over 6 divided by 5 over 6 cubed. So this is all they're doing in the problem. So you should be able to calculate the probabilities of this out between all of these, where the ratio or the percent chance of getting 1G to 1C, say you have a GCC sequence to a GGG, the chance of, or the probability of coding for this exact codon would be 5 over 6 times 1 over 6 times 1 over 6, divided by, of course, that 5 over 6 cubed. And for the last problem, the chance of getting all these cytosines is 1 over 6 times 1 over 6 times 1 over 6 divided by 5 over 6 cubed. You can just plug that into a calculator to see your chance for getting each of these. And of course, the other possible codons, such as GCG, will have this same ratio, because instead of this 5 over 6 here, you'd have the 1 over 6, and instead of the 1 over 6, you would have the 5 over 6. And you should still get the same probability, or the same chance of this occurring. For part C, they give us an amino acid table for this. And they are saying we have an equal proportion of cytosines and guanines. And they're asking us the percentages for getting each of the amino acids in these groups. So if you've never read from one of these tables before, you start on the left to check for the first amino acid. So we'll say we have a CCC sequence that we are looking to code an amino acid for. So you'll start with C, you'll move on to the second position up on top. So you move over from the table until you get to the column with the next C. And from there, you move on to the third position of the table, where you pull from the left. So if you do that for any amino acid, you should get the amino acid that is coded for by the codon. So for example, CCC would code for the amino acid of proline. So keeping that in mind, and the 1-to-1 ratio of guanine to cytosine, we should consider the different possibilities we have. We have a codon where we'll have 3 Cs to 0 Gs. We'll have codons with 2 cytosines to 1 guanine. We'll have a 1 cytosine to 2 guanines, and we'll have 0 cytosines to 3 guanines. And you can just write in the different combinations of these. So these should be some of the codons and DNA sequences that you get. And then from there, you can just use the table to figure out what amino acids these would code for. And you should get proline for the CCC, which I demonstrated above. You should also get proline here. You should get arginine, alanine, arginine, alanine, glycine, and glycine. And then you can just total these up to get a ratio. So we'll have 2 proline to 2 arginine to 2 alanine to 2 glycine. That can be coded for in this scenario. Of course, the percent chance of getting each of these is going to be equal because the chance of getting either G or C in each of these positions is 1 out of 2. So no matter what happens, you have a 1 out of 8 chance of getting any of these nucleotide or codon sequences. So you can just total these up and you should find that there are 8 parts. And because these are all equal and they have 2, each of these amino acids has a 1 out of 4 or 25% chance of occurring or being coded for when the G to the C ratio is equivalent. For the last part, we're being asked about our different stop sequences of, say, UAG. We also have UAA and UGA. When you look on the table and you code for each of these, you should see the stop inside the table rather than a 3-letter word for an amino acid or for methionine. The role of these is they do not code for any amino acid. So when we are creating our polymers of proteins and amino acids, instead of adding an amino acid to the chain, it's going to add nothing. So it's basically going to break off and end the protein sequence. So these are responsible for terminating the elongation of the protein and for finalizing it. So these are going to allow the amino acid chain and protein to dissociate from the ribosomes and to enter the cytoplasm in their complete form. Now with those 4, it should help you understand the purpose of the codon with its reliability and its 3 nucleotide sequence.